Hello, everybody, and welcome to this Genetic Engineering and Biotechnology News webinar. It's entitled Target Enrichment for Next-Generation Sequencing Analyses of SNPs, CNVs, Gene Fusions, and more. I'm John Sterling, Editor-in-Chief of Genetic Engineering and Biotechnology News, and I'm going to be serving as the moderator. Later on during the Q&A session, Talman Oliver, our Managing Editor, will take over the Q&A segment. Targeted resequencing is one of the fastest growing applications for next-generation sequencing technology. The ultimate goal is to look for causative mutations within discrete genomic loci in populations to enhance the development of new diagnostics and therapeutics. During today's webinar, a novel next-gen sequencing target enrichment method that utilizes single primer enrichment technology will be described and discussed. The approach has applications in the identification of gene fusion events in RNA samples, target gene expression analysis, variant detection, and copy number variation analysis in DNA samples. Our two panelists are experts in the use of this technology, so let's meet them. Dr. Stephen Kane is Director, Product Marketing for NUGEN Technologies. Dr. Jonathan Skolnick works as a scientist at the company. They're going to illustrate an innovative approach to variant detection and structural changes in DNA and to the detection of gene fusions and alternative splicing. Now, after the panelists have made their presentations, there's going to be a question and answer period. Feel free to send in a question for our panelists at any time during the webinar. Type your question into the Ask a Question box on the lower left of your console and then hit Submit. The panel will try to answer as many as possible. So if everyone's ready, let's get going. Dr. Steve Kane will be our lead-off presenter. Steve, floor's all yours.
Thank you so much, John. And as John had mentioned, the title of our webinar this morning is Target Enrichment for Next-Generation Sequencing Analysis of Single-nucleotide Polymorphisms, or SNPs, copy number variants, gene fusions, and more. So to begin with, I wanted to just go over the different topics that we'll be covering in today's webinar. And they're based on a related technology that NUGEN's developed for target enrichment. So the first area that we'll be talking about is related to our products for the Ovation target enrichment systems, which is a method for looking at variant detection and structural changes in DNA. A very related technology area and product area, the Ovation Fusion Panel target enrichment system, is looking at RNA-based analysis for the detection of gene fusions and alternative splicing. So before I get into these two different technology areas, for those of you that may not be familiar with NUGEN, I wanted to briefly go through some of our core technology areas shown on this slide. So NUGEN is a sample preparation company. We focus our product development and technology development on genomic sample preparation. And shown in this figure on the top, the target enrichment technology, this will be the focus of the webinar for today. Going around in the right-sided direction, we also have two different RNA-Seq sample preparation methods that we've developed. One is for single-cell technologies, so looking at very low input amounts of RNA for RNA-Seq analysis. We also have a very novel method for targeted transcript depletion that's used in another RNA-Seq sample preparation method. All of the systems that we'll be talking about today have a number of different options for multiplex sequencing, up to 96 different bar-coded libraries. But NUGEN also offers a custom automation service to work with a number of different liquid handling robotic systems to enable a range of our workflows for high-throughput applications. And then lastly, in the area of nucleic acid amplification, NUGEN has a number of technologies both for microarray applications as well as next-generation sequencing. In this central gold circle, simple and automatable assays, this is really a philosophy that we bring to all of our product development efforts, including the target enrichment technology. So we really strive to develop assays that are both simple to use and are able to automate as researchers begin to scale their projects. So now turning specifically to the target enrichment technology, I'm going to first show you mechanistically how this approach works, and then I'll also show you a stepwise protocol to see how it can be implemented in your research area. So the technology that is used in the target enrichment process is titled Single Primer Enrichment Technology, or it goes by the acronym SPET. And so to begin with, if you look at the top portion of this figure, this is representing how the technology would be applied to genomic DNA target enrichment. So you have genomic DNA with a hypothetical target enrichment or target region shown in the heavy blue rectangle. And in our process, we use something that we call a probe landing zone. And this is where we're going to design specific primers, in the case of genomic DNA, on the 3' side of the target region, interrogating each strand. So that's one important distinguishing feature of this method, is that we're able to collect sequencing information from each strand of the input genomic DNA or the input total RNA. So beginning with the process on the upper left, we start by fragmenting the DNA to a mean size of about 500 base pair. So you'll see that in this case you have your desired target region and the associated probe landing zone in the center of this figure. We then ligate an indexed forward adapter to each side of the molecules, and these are adapter sequences that are compatible with the Illumina sequencing platforms. And then the bubble number 3 on the upper right is really the key in terms of how the technology works. So the red arrow pointing to the left is a specific probe sequence that's designed to the 3' end of desired target regions. There's also a reverse adapter sequence shown in orange, which is part of that overall probe sequence. So we do an annealing reaction, as shown in step number 3, followed by an extension reaction with a polymerase coming off of the primer that is designated by the red arrow. We then copy all the way through the target region into the forward adapter sequence to complete that double strand. We then, in step number 5, denature and anneal PCR primers that are specific to the reverse and forward adapter sequences to expand those molecules. And then in the final step, number 6, we've generated an enriched library that's specific to the target regions designated by the different probes. So now to look at this same mechanistic approach now in a stepwise fashion shown on the next slide, we have the workflow for the Ovation target enrichment systems. So starting on the right side of this figure at the top, you'll see that the input requirements for fragmented DNA are as low as 10 nanograms up to 500 nanograms of input. As I had mentioned, we start by fragmenting to a mean size of 500 base pairs. We then do an end repair reaction, followed by adapter ligation, and then a bead purification. So this is about a 2.5-hour protocol with 45 minutes of hands-on time. We then primarily recommend an overnight probe annealing and extension reaction, which lasts about 12 hours. If there is a desire to have a rapid time to answer, we have an optional 3-hour protocol. So using that 3-hour protocol, you can complete this entire workflow in a single day. But more typically, we would recommend an overnight incubation of about 12 hours. On the second day, then, you would do a bead purification. There's an optional qPCR, quantitative PCR step. This is used to optimize the number of PCR cycles that would be used to expand the final library. After this step, then, you do the library amplification with the number of PCR cycles that have been determined by this previous qPCR step, a final bead purification, and then the result is a target-enriched library that's now ready for sequencing. So looking on the left portion of this figure, then, the workflow can be completed in less than 24 hours. That's 2 half days with an overnight incubation in between. It is a simple add and incubate assay, as I mentioned, automatable on a number of different platforms using various barcode configurations up to 96. So you'd be able to sequence 96 different libraries in the same sequencing run. And each of these adapter sequences are designed to be compatible with standard Illumina sequencing. I will briefly introduce you to some of the products that we've developed using this technology on the next slide, and these represent products that are used both for analysis of genomic DNA as well as for gene fusions using RNA. So the Ovation Fusion Panel is an RNA-based product. It contains content that addresses 500 different cancer genes. These are curated from the Wellcome Trust COSMIC database, as well as ChimerDB 2.0 databases, as well as from the different scientific literature that we've mined where there have been documented gene fusions. So using this product, you're able to look at both known and novel fusions, which Jonathan will detail in his section of the webinar. For DNA-based applications, we have the Ovation Cancer Panel. This is derived from 344 cancer-related genes that were reported in this publication in Nature Biotechnology. There's two different versions of this product, one that's optimized for good quality genomic DNA, and then another version that's optimized for more degraded genomic DNA samples. For example, those that will be found in formalin-fixed, paraffin-embedded samples. We also have another DNA-based product for the human mitochondrion. So this carries probes that are addressing the entire human mitochondrial genome. And then lastly, in addition to these standard products that have predefined content, NUGEN offers a custom target enrichment solution where we work with researchers that have content that they'd like to design using the SPET technology. And we produce custom kits that are configured to target regions of interest up to 10 megabases of size. So the total target region being up to 10 megabases. And this custom capability is true both for genomic DNA applications as well as total RNA applications for looking at gene fusions. So I'm now going to turn it over to Jonathan Skolnick, which will elaborate on the workflow again and show you various data sets related to both DNA and RNA applications. Before I turn it over to Jonathan, I would just like to remind you again that at the end of the webinar, we would certainly like to have questions from the audience, and you'll be able to enter those at the bottom of your console. So Jonathan, please take it away.
Great. Thanks, Steve. So I'm going to run through some key features of the assay, followed by some performance data from both genomic DNA inputs as well as RNA inputs, looking at the gene fusion detection. So I'm going to start by focusing on really some of the key features of the assay. And if you look at the figure at the bottom of the screen, this is a description of what your library molecules look like at the end of the assay, when they're ready for sequencing. So the DNA target here is represented by the thin black line. And everything to the left of that is ligated on in the first step. And everything to the right is brought in by the probe hybridization and extension. So if we focus on the left side of those target molecules, the real key feature that I want to point out here is that in the red box, we have an 8-nucleotide index sequence. So your samples are indexed in the first step of the process by ligating on this adapter. Those 8 nucleotides are followed by a random 6-nucleotide sequence. And this random 6-nucleotide sequence is used so that you can mark and identify PCR duplicates coming out of your assay. And to mark duplicates, we provide a software tool. And the way the software tool works is quite straightforward. So if you were to look at these 4 molecules we have here, the first thing the software will do is identify the start and stop positions of your DNA insert. Now, once 2 molecules have been identified that have the same start and stop positions, so for example here, the top molecule and the molecule on bottom, the software then goes in to look at the random 6 nucleotides and see if they're the same or different. So in this case, the top molecule has a gray 6-nucleotide sequence. The bottom molecule has a yellowish 6-nucleotide sequence. And so these 2 sequences are now known to be independent events, and they're not caused by PCR duplication. Now, if both of the N6 sequences were the same, then the software would mark those sequences as being PCR duplicates, and you could choose how you want to handle that in your later analysis. Now, the next feature I want to look at that Steve has already mentioned is the probe placement. So this is a key difference between our designs for genomic DNA or when you're using RNA. So as Steve had previously mentioned, when you're looking at a target region in genomic DNA, we place probes outside of that target region so that they extend into the target region, and that's what you sequence. Now, for looking at RNA, we don't have the intronic sequences to be able to target, so instead we place the probes inside each exon near an exon-exon junction. And you can see as the probes extend the direction that the arrow is pointing, the extension product goes through the exon-exon junctions, which captures information about splice sites or gene fusions. So for the next few slides, I'm going to walk through some performance data for genomic DNA inputs, and then that will be followed by gene fusion detection. So in this slide, showing data for a various range of probe sets that we've developed both in-house and for customers. So at the low end, we have our mitochondrial genome panel, which represents a 16 kb total region. And on the higher end, a test panel designed for a customer that was 7 megabases in size. And so what you can see is that regardless of the size of the target region, from 16 kb up to 7 megabases, the assay performs essentially the same. So we have a high percentage of reads that align back to the genome, a high percentage of those reads are on target, and the uniformity is relatively stable across those different target ranges. So in the next slide, we're showing some of this same performance data for both fresh samples and for FFPE samples. So the blue bars and the red bars represent the percent of bases aligned to the genome, or in the case of the red bars, the percent of those bases that are on target. And what we've done here is look at different input amounts from 10 nanograms up to 500 nanograms. And you can see that the assay performs essentially the same in terms of the percent of bases aligned and percent of bases on target, regardless of that input amount. The green line here represents the uniformity, and you can see in the fresh samples that the uniformity is roughly the same across the different inputs. And as you might expect, the orange line, which represents the percent of duplicate reads, decreases as you have more molecules available for the assay. And if you look at the FFPE samples, we see similar trends, although here the uniformity increases as you increase your input. Again, the duplicates decrease as you increase your input, again, because you have more molecules available for the assay. And in the next slide, we're looking at the effect of read depth on these different metrics. This is using fresh DNA. So if you look at the on-target and uniformity metrics, those remain constant across the different read depths. The percent of duplicates reads obviously decreases as you are doing fewer reads. And the percent zero coverage shows a very slight increase as you step down the number of reads you're looking at. But what I want to point out here is that even at only 70x coverage, there's fewer than 1% of your target regions that are not getting sequenced. And when you're increasing that up to 500x coverage, it's about 0.1% of the target regions that drop out. So when you think about your samples, not everyone has perfectly pure samples. You might be looking at a tumor that might be a mixed population of cells. And in that case, you may want to see what percentage of the cells or of the DNA from the cells can be identified with this type of assay. So to model that, we've taken 2 samples of DNA and we've mixed them at different ratios. And then we've gone back to look for the ratio of the alleles that we identify from the sequencing data. So you can see when we have a 50% mixture, we see approximately 50% of the alleles are derived from one sample and 50% from the other. At a 20% mixture, 20% of the alleles are derived from one sample, 80% from the other. And in the zoom-in box there, you can see that even down to 5% mixtures, we still see a 5% allele frequency And even at 1% mixtures, we see 70% of those alleles are still identified. So we're very confident that in mixed samples, you'll be able to identify at least down to 5% consistently with this assay. So that last slide was from fresh DNA. And in this slide, we're showing a similar type of metric, but using FFPE DNA. So this is data that was actually generated by a customer that they've allowed us to share. And it's using an FFPE sample from Horizon Diagnostics that has a known number of mutations with allele frequencies ranging from 3% to 33%. And if we look at those alleles, what we can see is that at either 50 or 100 nanogram inputs, we can get a 5% allele detection consistently across libraries. So those were some data for identifying single nucleotide variants. But we've also started to look into what happens when you have different copy numbers. And the way we've done that is shown on this slide. We took a sample that was a known trisomy 13 male, and we compared that to a wild-type female sample. Now, using our cancer panel, we had probes that were represented on each chromosome, and we then counted the number of sequencing reads from each of those probes, and we're able to identify the copy number of each of those chromosomes in both the trisomy and wild-type sample. And what you can see is that on chromosome 13, we have 335 probes that happen to land on chromosome 13. And it's very clear that you have a third copy of that chromosome in the trisomy sample. And we have 105 probes that cover the X chromosome. And again, it's very clear that the male sample only has one copy of the X chromosome. So now that I've shown data from genomic DNA inputs, I'm going to switch gears a bit and move towards our RNA inputs and fusion detection. So gene fusion detection is typically done by fluorescent in situ hybridization, FISH, or PCR. The problem with both of those assays is that it requires knowledge of both genes involved, and not only information about both genes involved, but you really need to know what the specific breakpoints are between those genes in order to detect the fusion event. More recently, people have been using standard RNA sequencing to be able to identify gene fusions, but as I'll show, that's a very inefficient method for detecting fusions that really requires millions of unnecessary reads. And where I'm going to focus our attention is on the Ovation fusion panel, because it offers a simple method for focusing the sequencing reads on desired gene fusion events. And what's important here is that it allows you to detect gene fusions even when only one of the two genes is targeted by our probes. So as Steve mentioned, we have a catalog panel of 500 known transcribed gene fusions derived from a handful of databases as well as the literature. And in this slide, I'm just going to show a sort of schematic like we've seen before, but focused here on how it is you can identify gene fusions from sequencing data. So if you were to start with your forward read, that read comes from the randomly fragmented end of the cDNA, and so it could be in any exon in any gene. So your reverse read comes from your targeting probe, and you know where that probe is sitting, and as you sequence through, that is your known exon that you've been targeting. And if you look at the red dashed line, that could represent an exon junction or a fusion site. So you can see that if your forward read and your reverse read are coming from two different exons or two different genes, or if one of those reads is long enough to actually read through that fusion site, then you can relatively easily identify that a fusion event has taken place. And here I'm just showing some examples of how we may be able to make those identifications. So in the case on top, we've targeted probes for both genes, both pairs of genes in this fusion, and you can see the green arrows on the top strands that are pointing to the left can be extended across into the red area, and there you would have your forward read would be from the red gene, and your reverse read would be from the blue gene. While on the bottom strand, the red probe can extend through to the blue area, and again, in this case, you'd have your forward read would come from the blue gene, and your reverse read would come from the red gene. Now on the bottom, I'm showing an example of where we only target one of the two gene pairs, and this allows you to identify previously unknown gene fusion events. So in this case, again, on the top strand, that green probe points over towards the purple and can extend into that region, which gives you a forward read from the purple gene and a reverse read from the blue gene. So we started our RNA sequencing fusion detection by looking at an H2228 lung adenocarcinoma cell line because we know that this cell line carries two gene fusions. And here is just some simple metrics looking at different inputs from 1 nanogram to 100 nanograms of total RNA. And you can see roughly 90% of the reads align back to the genome, with over 90% of those reads being on target. So in this cancer cell line, we are able to easily identify both of the gene fusion events, ALK-EMAL4 and ALK-PTPN. And these were identified using a piece of software called SOAPFuse. So once we were comfortable that we could identify gene fusion events in a single cell line, we challenged ourselves a little bit and turned to UHR RNA. So UHR is a mixture of 10 cell lines. So in this case, each fusion event that's detected is only derived from 10% of the total input material. And we know from the literature that UHR has a number of gene fusion events, and we happen to have targeted two of those events using an earlier panel that didn't have quite as many genes that were targeted. So we targeted two of the known fusions, and we were able to easily identify those, again, using the SOAPFuse software. In addition, we also were able to identify two new gene fusions that were previously undescribed in the literature. And the new gene fusions in this and subsequent data have all been validated by RT-PCR and Sanger sequencing. So we next looked at brain tissue, actually expecting it to not contain any fusions. But when we ran the assay, we were actually able to identify two. So now this brain sample is actually from a mixture of 23 individuals, which means that each event is only derived from 4% of the total material. And in this case, we identified the two fusions. The ATP5L-ML fusion is actually consistent with patients who have leukemia. And the FOXP1-EIF4E3 fusion is something that has not been shown in the literature up to this point. And the last cells I'll show actually were a tumor from a collaborator who had snuck in a positive control sample without letting us know. So when we sequenced the data and ran the analysis, we identified this COL1A1 PDGFB fusion, which is the hallmark of a particular tumor type. And we were able to go back to the collaborator and say, we think we found your tumor. And they said, well, yes, congratulations, that was our positive control. So we're fairly pleased about that. Now, this next slide is here to really show you why using targeted RNA sequencing for fusion detection is so important. So across the bottom, we have 4 fusion genes that we had identified in the UHR RNA. And on the y-axis, we're looking at the number of sequencing reads that indicate a fusion event. Okay. In the teal, we had a targeted library that was sequenced to 1.6 million reads. In the darker blue, we have a separate targeted sequencing library that was sequenced to a depth of 8.7 million reads. And in black, we have a publicly available data set that was a 125 million paired-end standard RNA sequencing reads. So what you can see when you look at the BCR-ABL is that even at only 1.6 million reads, we were able to identify the same number of fusion reads as in a data set of 125 million untargeted reads. If you look at the NUP214 XKR3 fusion, you can see at the 1.6 million targeted reads, we identify fewer than the untargeted sample. But what's important in this case is that we were only targeting NUP214. We did not have probes for XKR3. So you can see that you can still easily identify a gene fusion event when only one of the two genes is covered. And for the final two fusions, the FGF fusion and NLLT10 fusions, we were unable to identify any sequencing reads in the untargeted data set. So out of 125 million reads, you still weren't able to identify these gene fusions that we were able to detect with a fraction of the number of targeted reads. So this slide is a schematic of our 500 gene fusion panel and just shows all of the known fusion events that you might be able to identify using this panel. And I'm going to stop here and summarize what I've talked about. So the Ovation target enrichment product creates targeted sequences from libraries with as little as 10 nanograms of genomic DNA or total RNA inputs. And it works with both fresh DNA as well as FFPE samples. We have a fast, flexible, custom probe design with low dropouts, low design dropouts, as well as very low experimental dropouts, which I showed previously. Our enriched libraries exhibit a high percent of reads aligning back to the genome, with a high percent of the bases aligning back to your target regions of interest, as well as even coverage across those regions. And with the fusions, I showed how by using a targeted gene fusion assay, we're able to detect more gene fusions than using many more reads from a standard RNA-Seq experiment. Now, for the gene fusions, we have a catalog panel of 500 cancer genes, and both for the gene fusion detection as well as the genomic DNA targeted sequencing assay, we have custom panels available, and that is a 3-week turnaround time from the customer design approval to when you get it in your hands, ready to get your lab work done. And so with that, I'd like to thank you for your time and joining us today, and I'll hand it back to John to take any questions that you have.
Well, thank you, Jonathan, and thank you, Steve, for both of those detailed and timely presentations. As I mentioned in my introduction, targeted enrichment and next-gen sequencing are two of the most fertile areas in biotech R&D going on right now, so thank you. You both provided excellent insights and advice on how our webinar attendees can carry out their own operations in these areas as efficiently and economically as possible, so again, thank you so much. We're going to begin our Q&A segment very shortly. Feel free to send in a question for either of our panelists. Type your question into the Ask a Question box on your computer console, and then hit Submit. But before we start the Q&A, I'd like to ask you to disable your pop-up blockers. A short survey on this webinar is going to be appearing in a moment. We would very much appreciate your feedback on the webinar. At this point, I'm going to turn the webinar over to Tammy Oliver, Gen's managing editor. She's going to handle the Q&A. Tammy?
Thanks, John. Our first question is, what is the limitation on size of custom captured territory? Why only 10 megabytes?
So this is Steve. I'll take that one. There isn't a technical limitation on the size. We've done, as we discussed during the webinar, we've done capture work down to a few kilobases. We've done experimental work up to 11 megabases internally, and we believe that we can go higher in terms of the upper limit of the target region.
Okay, another question is, which Illumina platform would you run this on?
This is Steve. I'll take that. We've generated data on a range of different Illumina platforms, the MySeq, the Genome Analyzer 2, as well as the HiSeq platform. So the chemistry in terms of our library construction workflow is compatible with the Illumina sequencing primers that are used across all of their platforms. So we don't believe there's a restriction there.
Okay, here's another question. Can non-model organisms be used for the custom target enrichment solutions, i.e. customer supplies target region in bed format and reference genome in FASTA?
Yeah, so this is Jonathan. That's absolutely something we can do. We can use any FASTA sequence that you provide, and as long as you can also provide us the regions that you're interested in targeting. We've done, I think, while we've focused generally on human, we've done mouse, rat, a couple of different plants, and some viruses. So we have the pipeline set up to be able to handle that pretty easily.
Okay. Can we define a custom RNA panel, for instance, from the human RefSeq database?
Yes, this is Steve. So that's really core to the product offering that we have, and that we do have a few products, as we discussed, with predefined content. But we're very much set up to work with researchers to design custom panels, both for RNA workflows as well as DNA. So absolutely, the answer is yes.
Okay. Did you try your technology on WGA DNA? If yes, how well does it work compared to bulk DNA?
So this is Jonathan. We've had customers use WGA to input from single-cell levels of DNA, and then they've fed back the data to us. And what we found is that the assay performance is essentially unchanged in terms of the on-target performance. But what happens is the evenness of coverage decreases, basically, because the amplification itself adds some bias into which regions are amplified.
All right. What is the lowest input level that you would recommend for FFPE samples?
So this is Steve. We did discuss some data using FFPE during the webinar. Our stated minimum input level would be 10 nanograms for FFPE samples. But you will note that the duplication rate and the uniformity is improved at higher levels of input. So we would say that if you have samples available to go to higher levels, say in the neighborhood of 50 nanograms, that would be preferable. But the minimum input level is 10 nanograms.
Okay. Do you provide software for the PCR replicate detection using N6 tag?
Yes, so we do provide a script that can be used. Basically, you would take your data, map it back to the genome, and then apply this script that we provide, which will go in, look at the mapping coordinates, identify those reads that have the same mapping coordinates, and then check that random 6-mer sequence to see if they're the same or different. If those random sequences are the same, the software then marks those as PCR duplicates. And from that point, you can decide in your own analysis how you want to handle that, whether you want to just eliminate those duplicate reads, or you want to use them in some of these newer informatics approaches that have been described in the literature to use those duplicate reads to identify errors from the actual sequencing instrument.
All right, and how many sequencing reads do you recommend for the 500 gene fusion panel?
So for that panel, we would recommend about 8 to 10 million reads. That will get quite a comprehensive view of what's happening inside your samples.
And which human brain reference did you use?
So the human brain reference was an Ambion product, which is a mixture of 23 different brain samples. It's something you can just purchase directly from them.
Okay, unfortunately, we've run out of time. Thanks for listening in today, and thanks for the great questions. This webinar will be archived on our website for 6 months, www.genengnews.com. If you missed parts of it, you can watch it again, or you can recommend it to friends and colleagues. Once again, I want to thank our panelists as well as our sponsor, NUGEN.